quick, very warm welcome from myself. My name is Tutu, head up the RNA Leaders team at LSX, and great to have you all join us here today for our post RNA Leaders Europe webinar with our hosts. Just a quick thing before I hand over to a host and speaker. This session is interactive, so we encourage questions. To do that, we ask that you ask any questions in the Q&A chat function, which is at the bottom of your screen. If you have a question, we encourage you to do it, think about and do it straight away, just so great. Just so is at top of mind, so you kind of ask at that point. You also can upvote any questions that you see. If someone's asked a similar question that you're thinking about, upvote it so that will be asked first. And if any comments in terms of to be able to expand a question, feel free to comment on the questions that are there as well. So yeah, we do have at least 10 minutes for questions at the end. So yeah, we do kind of want to get to that discussion point at the end of the session. So yeah, I think we've got quite a few people have joined, which is great to start kicking this off. And I'm going to hand it to Birgit to kick off the session. Thank you.
Thank you, Jade. A very warm welcome to the webinar from Roche CustomBiotech with RNA Leaders and our speaker, Axel Guilbaud from Genentech. And it is a great pleasure to know that so many people from all over the globe dialed in today. So very exciting, because it's also an exciting topic on very advanced and new technologies for the comprehensive characterization of mRNA and its impurities. So really a burning topic in the field. My name is Birgit Rogell, and I'm Regional Product Manager for mRNA raw materials at Roche CustomBiotech EMEA LATAM. I will guide you through today's webinar, and then I hand over later on to Axel. So let me first give a short introduction about Roche CustomBiotech. We are hosting today's webinar before I hand over to Axel, as I said. As part of Roche, CustomBiotech has been a leader in supplying innovative, high-quality products to forward-thinking biopharma and diagnostic businesses for more than 50 years. Our pioneering biopharma and diagnostic experts work together at one of Europe's largest, most productive biotechnology centers in Penzberg, Germany. So here you can see the site in Penzberg, Germany, very green out there. One reason for our success is our world-class facility in Penzberg, Germany, the Center of Excellence for R&D and Manufacturing, and one of the largest, most productive biotechnology centers. And it has an unusual setup that merges Roche diagnostics and pharmaceutical know-how under one roof. We offer a broad portfolio of materials along the manufacturing workflow of diagnostics and biopharma companies, like our mRNA raw materials portfolio that you see on the left-hand side. We offer mRNA raw materials in high quality and scale, ready to use in the manufacturing of mRNA vaccines and therapeutics. We just launched our nucleotides in animal origin free and in GMP grade. So you can still reach out to us and ask for test samples. So contact us after the webinar to learn more. Our production is based on green chemistry, which is nice for the ecological footprint, but also nice with regards to the reduction of impurities usually associated with the chemical synthesis. So thank you for your attention. If you go on our website, you can dig deeper into our portfolio, different portfolios, and also learn more about the quality, IVT reaction conditions, etc. Now to our today's speaker. Hello, Axel. I'm very excited to welcome Axel Guilbaud. For him, it's very early morning to join this webinar, so because he is joining from Genentech in San Francisco. Axel is principal scientist at Genentech, whose expertise lies in mRNA and gene therapies. And he is working on the comprehensive mRNA characterization because it's indeed a burning topic. And he tries to dig deeper and advancing the technologies to really learn more about how to solve the knowledge gaps in this field. And I can already say it's very new and exciting what he has to share today. And I'm very much looking forward to see his presentation, but you can also see his presentation by going, and there is a recent publication that is already out, so you can check PubMed on analytical chemistry. So please, Axel, I hand over to you and looking forward to your talk.
Yes, thank you for this introduction. Let me just share my screen. All right, so hi everyone. So I'm really happy to be here to discuss about the work that we did on mRNA and trying to characterize these molecules and its impurities, especially by using some current and novel analytical techniques. So before, I just want to give you an introduction on the state of the state of the gene and theme therapies. So it is briefly composed of three main fields. So you have the RNA therapy containing the mRNA vaccines, therapeutic, short interfering RNA, antisense, and other non-coding oligos. You also have the gene therapy with the viral and non-viral gene therapies, gene transfer, and the last one is the cell therapy containing the non-gene modified cell therapies. So these three fields are interconnected, and there is a growing interest in developing preclinical and clinical trials by using these cell and gene therapies. So in 2023, there were over 4,000 of these preclinical and clinical studies. Why do we have an increase of the interest in that? It's because there is an improvement of the technological advancement and innovation, so especially for gene editing technologies such as the CRISPR-Cas9. But you also have an improvement in the manufacturing process and also in the delivery methods that have played a crucial role in the growth of cell and gene therapies. As I just showed you, there is more and more clinical successes and regulatory approvals with, for example, the CAR T cells. And because of that, there are also more and more investment and funding that's enabling the translation from research to clinical application. So what actually can we do with RNA drug therapies? So there are several applications with this first one, which is the therapeutic protein production. So here, for example, if a patient is not able to produce an active protein, here you can incorporate the mRNA in the cells of these patients and generate some active proteins. There are also protein replacement, where here you want to restore the normal physiological function of a specific non-functional proteins or receptors. You also have the mRNA-based vaccines applications. Here it's more to prevent against infectious diseases and treat it against some type of cancers. But mRNA can also be used as a critical raw material for genome editing with the CRISPR-Cas9 strategy. And the last application is the cell therapies with the CAR T cells, where here you are going, for example, to collect the lymphocyte T of a patient, and you are going to introduce an mRNA that will help to produce some receptors on the surface of this lymphocyte T. So there are other advantages of the mRNA drugs therapies. So, for example, you can theoretically produce any kind of protein and peptides using the protein synthesis machine of the transfected cell. There is also proof in the literature that the mRNA drugs therapies can exhibit a high transfection efficiency and a low toxicity compared to the DNA-based drug. And this is because the mRNA doesn't need to enter in the nucleus to be functional. Another key concern is the potential mutagenesis for DNA drugs therapies. But here with mRNA, you're not going to integrate the host genome, which is reducing the risk of insertional mutagenesis. I also discussed previously about the different applications that you can have by using mRNA, but there is also, last but not least, advantages of the mRNA. It is that this technology, so designing an IVT mRNA, is straightforward, which allow for rapid, scalable, and cost-effective production. And we saw that, especially during the pandemic, the COVID-19 pandemic, that there is a real, we can face an emergency and where a rapid vaccine development and a large scale production are paramount. So after the application and the advantages, how can we basically create some IVT mRNA? So I will go briefly about that, and you will see that IVT mRNA, so in vitro transcription, can have its own set of challenges. So the first step is basically to mix the DNA template with the nucleotide triphosphate and the T7 RNA polymerase, and then quench the polymerase reaction by using some enzymes. So from here, you will get a bulk materials with your unpurified mRNA. And after that, you will have several steps to be able to purify it. So these steps are time consuming and they need to be performed accurately, because otherwise that can lead to some mRNA degradation and also reduce the quality and the yields of your mRNA. Because mRNA are inherently unstable and they can go under hydrolysis during the upstream and downstream manufacturing process. So also for the preparation, the processing, the formulation, but also for the long-term storage. So that's why there is an imperative need to characterize mRNA and also the different impurities that can be created during the different purification steps. That's really important if we want to ensure the quality, understand the activity, and also in the future, optimizing the manufacturing processes. mRNA have unique properties, so they are polar molecules and they are highly negatively charged. They contain secondary structures, but also sequence features, such as the 5' cap, which is present at the beginning of every eukaryotic mRNA. And the role of this 5' cap is basically to promote the translation initiation and also to promote the mRNA stability by avoiding its degradation through nuclease. The 5' UTR for untranslated region is a non-coding sequence before the open reading frame. And it has a crucial role because it's going to bind the ribosome, which will initiate the translation. So it's also playing a role in mRNA stability and trying to modulate the translation initiation. The open reading frame is basically containing the sequence in your mRNA that is going to be translated into the active proteins. The 3' UTR as the 5' UTR are untranslated region as well, and they are going to help to determine the mRNA half-life, promotes the mRNA stability, and also regulates the translation. The last part of this mRNA structure is the poly A tail. So basically, when to create an IVT mRNA, there is an addition of 90 to 120 adenosine at the end to improve the stability of the mRNA. But this poly A can also bind to the ribosome, which will also improve the translation step. You can also do, you can also modify some of the nucleotides. So for example, by adding some methyl group. And the purpose of this modified nucleotide is basically to enhance the mRNA stability, but it can also decrease the mRNA immunogenicity and also protect against the degradation from nucleases. So as you can see here, there are many features that can be pretty much important, and any disruption in one of them can lead to the formation of fragmented RNA molecules. And at the end, if you are, for example, missing the poly A tail, at the end, the mRNA will not going to enter in the cells, and consequently, the mRNA will not be able to perform its intended function. So be able to characterize all of these different critical quality attributes that can have an impact on the safety, the immunogenicity, the bioactivity, and the pharmacokinetics. There are some there are some experiment instrumentations and methods that existing to help characterize them. For example, if you want to monitor the integrity and the purity of your mRNA, the traditional methods are the gel electrophoresis, the fragment analyzers, and the ion pairing reverse phase liquid chromatography. To test the identity of your open reading frame and be sure that your sequence is full, that there is no missing or additional sequence. The method of choice are the sequencing, so with the Sanger or the new generation sequencing, but also as for antibody, you can use the oligo mapping by LC-MS-MS. The LC-MS methods can be used also to do some quality testing, especially the capping efficiency and the poly A tail. And as you can see here, there are some other parameters that can be important to monitor, such as the content of your mRNA by RT-qPCR or ion pairing reverse phase liquid chromatography, but also some functionality tests. So here it will more depend on what you want to do with your mRNA, if it's more to use this mRNA as a critical raw material, or if it's more for mRNA vaccine, for example. So here, there are different in vitro translation assay that can be performed, but also Western blots and cell-based assay. A last part is the impurities, which are also important and that can be formed during the manufacturing processes. And one of the most famous one is the double-strand RNA that can be increased during the IVT reaction. But you also can monitor other impurities such as the residual DNA, the proteins, the NTPs, and solvents such as the ethanol and the isopropanol. So, like antibody, since many decades, there is a huge need to develop several analytical methods to be able to cover all type of impurities, the size variants, the structural variants, because all of these mRNA impurities that can be created during the manufacturing processes and that can have an impact on the mRNA structure and its function has been largely unexplored. So there is a huge effort to try to develop these analytical methods. And as Birgit said, we just submitted a paper a few weeks ago based on the evaluation of traditional and advanced technologies to be able to characterize the mRNA and its impurity. So that's what I'm going to start to discuss right now. And I first want to introduce what we did to assess the purity of the mRNA by using the ion pairing reverse phase liquid chromatography, but also the capillary gel electrophoresis, which are the traditional methods. Before going further, I just want to give you two important information. The first one is that compared to antibody, for which the size is basically between 100 and 150 kilodalton. Here, mRNA size are way more bigger. So it's between 200 over 1,000 kilodalton. So for the ion pairing reverse phase liquid chromatography, we needed to work with the size pair of the column that were bigger compared to the column that are used for antibodies. The other parameters is that the mRNA are negatively charged, and if we want to improve the if you wanted to improve the separation, the resolution of the separation of the mRNA, we needed to work with an ion pairing, so the TEA. And basically, this TEA, which is positively charged, is going to bind to the T18 surface of your column, and then the mRNA are going to bind to this positive charge through their negative charge, which will improve the resolution of their separation. So this is what we did here on three different GFP mRNA. So, I mean, three GFP mRNA from three different vendors, so vendor A, B, and C. This GFP mRNA is about 980 nucleotides, and it's about 350 kilodalton. So as you can see here, we observed a typical purity profile of the mRNA with 3 different regions. So the pre-peak in blue, the gray peak in the main peak in gray, sorry, and the post-peak in red. The purity profile, as you can see, and this is also important to note, are pretty different between each vendor, and this is highlighting the impact of the manufacturing process, so the upstream and the downstream, on the mRNA quality. In parallel, we also wanted to use another traditional method, which is the TGE. So basically, here you have an anode, a cathode, and there is a capillary between the anode and the cathode. And your mRNA that are negatively charged, they are going to reach the positive side when you're going to apply a voltage. So here you will be able to separate your mRNA according to their size. And here, as you can see on the right, the CGE exhibits the same 3 regions as demonstrated by IP-RPLC. So we still have the pre-peak, a main peak, and also a post-peak. So the level of the post-peak impurities are slightly lower with capillary gel electrophoresis, mainly because there is a better separation of these variants compared to the IP-RPLC, as you can see here for the GFP, where the post-peak is way more resolved compared to the IP-RPLC. The other thing is that the improved resolution in CGE was also attributed to a decreased level of the mRNA secondary structures due to the stronger denaturing condition for CGE compared to IP-RPLC. So unfortunately, when you are taking this set of data, there is no clear information of the identity of this impurity. So we have a pre-peak, we have a main peak, we have a post-peak, but what does that mean, and what can we have in these different regions? So this is what we wanted to know, and we started to focus on the pre-peak, especially by developing 2 in-house IVT mRNA with the exact same open reading frame and the exact same structural features, except that for one of them, there was the polytail, and for the other, the polytail was absent. So in blue, this is the mRNA without the polytail, and in orange, this is the mRNA with polytail. And what you can see here, especially if you're taking a look on the IP-RPLC here, is that the blue trace here has a retention time that is matching perfectly with one of the peak that we are observing in the pre-peak region here. So meaning that one of these pre-peak that we have in the 3 region correspond to a poly-A tailless variant. You can also see that there are some additional peak here between the polytailless variant and the mRNA main peak with the polytail, meaning that here this peaks can be attributed to abortive transcript in the polytail sequence. So an mRNA with a polytail, but with a shorter polytail. When you're taking a look on the CGE LIF here, you can see that the blue trace here, corresponding here again to the mRNA without the polytail, was shifted by 3 minutes compared to the mRNA with the polytail, demonstrating that CGE can separate according to the length of the mRNA. After that, we also wanted to see what would happen if we would stressing the sample. So we stress the sample at 40 degrees for 5 days. And these are the strip line that you can see here and here. And if you're comparing the strip line with the plain lines here, you can see that you have a huge increase of this pre-peak here, corresponding actually to the poly-A tailless, meaning that upon heat stress, the poly-A tail is mainly degraded. So we know that the loss of the polytail can have a negative impact on the mRNA functionality, because without a poly-A tail, the mRNA can barely enter into the cells, and that means at the end you will have a lower amount of your proteins that will be expressed. So these data are actually showing the importance of monitoring the prey region, especially during stability studies. So after that, especially for the CGE, we had some internal discussion, and we had another technology which would be potentially used as an alternative for the traditional CGE, and this is the microcapillary electrophoresis. So here we are comparing the CGE, the traditional CGE, with the micro-CE under denaturing condition. We analyzed 3 different samples, a GFP, Fluke, and Betagal. So just to give you an idea, the length of a GFP is about 1,000 nucleotides. For Fluke, it's more 2,000 nucleotides, and for Betagal, it's 3,500 nucleotides. And with these 2 different methods, we were able actually to separate these 3 different mRNA according to their size. The most important factor that differ between these 2 technologies is the separation time. Actually, for CGE, the traditional one, a run can take more than 62 minutes. For micro-CE, it's more 1.5 minutes, which is drastically reduced. Other advantages of this technology are that the sample preparation time is lower, the amount of sample that you need is lower as well, and it is a high throughput technologies because you can run more than 300 samples in a row. But now let's focus on the profiles that we are getting from these 2 technology, and let's see if we can have like the same trends or not. So here are represented the percent purity of the 3 different mRNA with the 2 different technologies. So the white bar is the pre-peak, the black bar, the main peak, and the gray bar, the post-peak. As you can see here, the trend between CGE and micro-CE are almost identical in terms of percent purity. So if you're taking a look of the GFP, for example, here, we have like 60% for CGE for the main peak, and we also have 60% for the main peak in the micro-CE. So basically, the micro-CE can demonstrate the same sensitivity and the same separation resolution compared to the CGE, but in a shorter time. Another advantage of the micro-CE is actually that you can run NCE in both denaturing and non-denaturing condition. Why it's also important to run in native non-denaturing condition? It's because you can also track some information that you can miss by denaturing, especially for secondary structures such as non-covalent aggregates. So there are previous data that confirm that the functional activity of single-guide RNA, so a critical raw material for CRISPR-Cas9, is mainly due to monomeric forms in vitro. And the level of aggregates should be monitored since they can have an impact on the efficiency of your RNA molecules. And the best method to do that, actually, and the most famous one, is the size exclusion chromatography. But here, because we can also run micro-CE under native conditions, we wanted to compare these 2 technologies and see if one of them can be, if the micro-CE can be used as also an alternative for ATC. So as you can see here, we also analyzed the 3 same mRNA, a GFP, Fluke, Betagal, and at the bottom, you can see the percent purity for micro-CE on the left and SEC on the right. The first thing that I just want to let you know is that micro-CE and SEC are like working in mirror. So let me be more clear. So for micro-CE, you are going to separate according to the size. So they are going to move through a capillary. So the low molecular weight are going to be on the left of your egram, and the high molecular weight are going to be on the right side of your egram. For SEC, it's the opposite, because the biggest molecule are going to leave the column before, so they will be appearing on the left of your chromatogram, while the small molecules are going to interact with the pore of your column, and we leave the column at the end. So the short and the low molecular weight will appear on the right of your chromatogram. So here, as you can see, under native conditions, both MCE and SEC consistently indicated the presence of aggregates for the different mRNA. And you can also see that the percent purity is almost similar between micro-C and SEC. So for example, for the GFP here, we are close to 55-45% of main peak and aggregates. And for SEC, we are also close to 60 and 40%, which is pretty close. So after that, we also wanted to analyze the samples after a denaturing condition. So we heated the samples at 70 degrees for 10 minutes. And what we observed, especially if we are focusing first on the DFP, is that these aggregates that you could see here under native condition almost disappeared in denaturing condition. So we demonstrated that we were able to reduce the level of mRNA aggregates under heat step, meaning that those aggregates mainly corresponded to non-covalent aggregates. As you can see here for OGFP, we only had at the end, after heating, 5% of aggregate. So as you can see here, micro-C can provide almost the same information as the traditional CGE and SEC. But one limitation of the micro-C is that if you have big molecules, and we observe that with beta-gal, but also with bigger mRNA, you can decrease the resolution of your separation, and it can start to become hard to separate the different aggregates. For SEC, the limitation is also associated with the size of the mRNA, because there is a limitation in the size pore of your column, meaning that you will also have, with big molecule, a low separation resolution of the different types of aggregates. So to try to bypass that problem, there is another technology that I want to present to you, and this is the mass photometry. So it's basically working pretty similarly to a nanodrop. So you have to prepare your sample and put it in a glass, and then through a light scattering phenomenon, a laser is going to analyze your different mRNA. So it is an automated and user-friendly instrumentation. It's a quick assay, no more than 5 minutes per sample. And also an important factor is that the sample preparation is super easy, because you just have to dilute your samples in PBS or TE. In an interesting way, this instrumentation can also exhibit an accurate mass calculation, and the baseline resolution between each species, so monomers, dimers, trimers, is also robust. Compared to some technologies such as the SEC moles, so moles is another technology that is used to also characterize aggregates. Here, mass photometry doesn't require a large amount of samples. And the huge advantage is that you don't need here any column. So you don't have this pore size limitation here that you can have with the SEC column. So regarding the data, so here are two different mRNA. So the in-house mRNA. So the blue one is the mRNA without the polytail, and the orange one is the mRNA with the polytail. On the top of the monomers that you can see here, this is the length of the mRNA that was calculated by the instrumentation. And the interesting thing is that the difference between the theoretical and the experimental length was pretty fine, because the RSD percent values between the theoretical and the experimental were below 3%. Then we also determined with this instrumentation that the difference between these two mRNA, so the without and with polytail, was actually corresponding in a precise way to the loss of the polytail, which was about 100 nucleotides difference. The third parameter is, as I mentioned, with this instrumentation, we are also able to determine some species such as dimers, trimers, and you can see that the baseline resolution is really good between the monomers and the dimers. The interesting thing is that there are higher amount of dimers for the mRNA with the polytail, which is 16% compared to the mRNA without the polytail. So we were thinking that those dimers were considered as non-covalent. Why? It's because they were not present in the profile of other technologies such as the ion pairing, reverse phase chromatography, and CGE, because those technologies are based on denaturing condition, while the mass photometry is under native condition. Then we also decided, as previously, to heat the sample. So here at 70 degrees for, 40 degrees for 5 days. And what you can see first is that the dimers almost disappeared, which leading to the conclusion that actually those dimers were non-covalent secondary structures. And the other interesting thing that we observed here, a huge increase of the pre-peak, so 34% and 37% respectively, for the mRNA with and without the polytail. This is also something that we saw, if you remember, in the first data that I presented to you for the ion pairing reverse phase chromatography. We also observed some degradation in the pre-peak. So here with mass photometry, we also saw this kind of degradation. So after that, we also wanted to see if the mass photometry could be applied for bigger molecules. And here we used GFP, Fluke, and beta-gal. Just to remind you, a GFP is 1,000 nucleotides, Fluke is 2,000, and beta-gal is 3,500 nucleotides. And here, as you can see, again, the difference in the length of the monomers, the difference between the theoretical and the experimental was really low. I mean, below 3% in terms of RSD. So the thing is that the MP was able to actually determine in an accurate way the different monomer, even of big molecules. And it was still able to have a good baseline resolution for big molecules because we were still able to determine the apparition of some dimers in here for some dimers and trimers for a GFP. You can also see here that it highlights the difference in the process because we do not have the same amount of dimers and trimers in the different mRNA or different vendor here. So actually, the mass photometry technology provides valuable information, particularly for the detection of non-covalent species. But if we have non-covalent species, that means we also have covalent species. So to analyze the covalent species, we step back to the microCE and IP-RPLC because those technologies can be used in denaturing condition. So you have microCE on the left and IP-RPLC on the right. And here we used the same mRNA, so a GFP. And I also highlighted here the aggregates here, so the double-strand RNA here for IP-RPLC and microCE. And what we can see here that under denaturing condition, both microCE and IP-RPLC consistently indicated the presence of covalent aggregates. Covalent aggregates because we are in denaturing condition, and because we are in denaturing condition, all secondary structures that are non-covalent are disappearing. In literature, it's known that some of the covalent aggregates can be associated to 3' extended loopback formation during the IVT process. And it has also been demonstrated that the adoption of some alternative polymerase can have an impact on this 3' extended loopback. So here on this figure, you can see that we use 3 different T7 polymerase, so T7 wild type 1 and 2. And actually, when we use this alternative polymerase, if you're focusing on the gray part here that is corresponding to the aggregate, we observed a decrease of this covalent aggregate, meaning that we can actually do the association between the post-peak and this undesired byproduct. We observe this trend in both microscopy and reverse phase liquid chromatography. In parallel, we also use dot blot, which is actually an alternative tools to detect the double-strand RNA. And here again, so you have the T7 wild type on the top and the T7-1 and T7-2 at the bottom. And you can clearly see that with this alternative polymerase, there is a reduction of the double-strand RNA. Sorry. So now that we describe what we can have in the pre-peak and what we can have in the post-peak and the aggregate, we also wanted to go further and just try to do some extended characterization by using mass photometry and see if we could have additional information on the pre-peak or the post-peak. So just to give you some context, there are some studies that are using the mass photometry, but they are more to monitor some critical structural features such as the poly-A tail length and distribution and the 5' and capping efficiency. So if you want to analyze this kind of critical structural features, you need to digest your mRNA. That means that those studies are using non-intact mRNA. The few studies that have reported the use of mass spectrometry for intact mass were not satisfactory enough to get an accurate determination of the molecular weight. So there are challenges. So the first one is that the major heterogeneity of mRNA mass profiles is related to the poly-A tail distribution. And the second thing is that, as I described previously, if we want to separate chromatography talking, we need to use an ion pairing. So if you're using an ion pairing, that means you need a dedicated LC-MS instrument. So there is one solution, which is the use of the UHMR mass spectrometry system. And this instrumentation is theoretically applicable to any size, complexity for mass analysis. And it is fast analysis. It required a low amount of samples. And the advantage is that you do not need any HPLC column. So you don't care anymore about the ion pairing. And here you are able to analyze your mRNA under native condition. So to try to do some analysis, we took the mRNA. So still as the same as previously, we have the mRNA without the polytail and the mRNA with the polytail. And here is what we obtained. So first, for the intact mRNA here without the polytail, you can analyze here, you can observe that there is only one peak here corresponding to the mRNA of the exact mass of 224,000 Dalton. When we are analyzing the intact mRNA with the polytail, you can see that you have a strong heterogeneity of your signal here. And when you are zooming in, you can actually see that this difference between each peak correspond to 330 Dalton, which is actually the length of the molecular weight of the adenosine. But we still needed to prove that this heterogeneity was due to the poly-A tail distribution. So to do that, we digested the mRNA in order to get the poly-A tail alone and to analyze it. And here are the data that we get. As expected, there is a huge distribution of your polytail. You can see that the length of this polytail was between 96 adenosine to 105 adenosine. And this actually explains why there is such an heterogeneity here for the intact mRNA with the polytail. Then to go further, we wanted to trying to do some offline fractionation by using IP-RPLC. So this is a profile that we get from the GFP mRNA. And the first thing that we did was to first fraction collect the main peak and to analyze this main peak. And here you can see what I just discussed previously. There is only one signal here with a huge heterogeneity that is due actually to the poly-A tail distribution as described previously. Then we also did some offline fractionation on the pre-peak. And interestingly, here we observed two main impurities. The first one is actually the mRNA without the polytail. And you can tell that because if you're taking the mass of the main peak here, which is 176,000 Dalton, and you are subtracted this main peak here of the mRNA, which is 141 kilodalton, you are actually obtaining the exact mass of the polytail. So this one was attributed to the mRNA without the polytail. The second one here is more related to an abortive translation in the poly-A tail sequence. If you remember at the beginning of the presentation when I showed you the IP-RPLC, especially with and without polytail, I showed you that there were some additional peak between the polytailless and the main peak. And here actually we demonstrated by mass spectrometry, sorry, that there was actually a peak that was corresponding to the mRNA plus this a fraction of the poly-A tail. So just to conclude now, I just wanted to let you know that we presented here in this presentation the different technology that we used to characterize the mRNA and its impurities. So from the traditional one, IP-RPLC and CGE, but also some novel technologies with the native mass spec, the mass photometry, but also the micro CE that can be used as an alternative of the traditional CE. And the first thing to note is that the nature of pre-peak impurities. So with ion pairing reverse phase liquid chromatography, capillary gel electrophoresis, and native mass spec, we were able to demonstrate that pre-peak impurities correspond to the mRNA variants, so the polytail-less or with different polytail-less coming from aborted addition or partial hydrolysis. So these data show the importance of monitoring actually this pre-peak region, because we know that the loss of this poly-A tail can have an impact on the mRNA stability and its efficiency. Then for the investigation of the aggregates, so micro-CE and IP-RPLC methods, especially in denaturing conditions, have demonstrated their ability to characterize the covalent 3' loopback form. SEC also demonstrated the ability to monitor the aggregates in their native forms, so non-covalent plus covalent aggregates. And the mass photometry was also helpful to determine the presence of non-covalent aggregates, transcripts, and especially the detection of dimers, trimers, and even tetramers sometimes. So for this work, I really want to thank all of these people that are here, because that's, as I said, this was like a really huge team effort, and from different departments in our company. And this is, yeah, always really exciting to work with talented people, and it's really, really fun actually to work with all of these people and doing all of these jobs. So now, if you have Q&A, if you have questions, sorry, I will try to answer them, but just also wanted to let you know that if we do not have time, here are the email address, and you can still contact us in the future. So thank you for your attention.
Thank you, Axel. I mean, you're saying if there are any questions, many questions came in into our Q&A, and I can motivate people. If you have more questions, enter them, and we see how far we get. Very diverse tool set and very exciting technologies you share, especially also going into poly-A tail analysis. So for poly-A tail, there have been several questions, especially when you mentioned the degradation of poly-A tail. So are you sure that this is degradation, or can this be also oxidation? Or so what really leads to this kind of shortening of the poly-A tail that you observe?
So actually, for the poly-A tail, it can be whether a degradation. So because we know that it can be subjected to some hydrolysis, but it can also be due to, during the processing, the manufacturing process, there are different ways to add the poly-A tail. So the first step is directly to add this poly-A tail in the DNA template. And the other way is to add this poly-A tail enzymatically. And sometimes also by doing enzymatically, I guess that it was demonstrated in the literature that you can have a partial integration enzymatically, and your poly-A tail is also less stable when you're adding this poly-A tail enzymatically at the end. So this is the degradation of the polytail can come from directly in the process because it's not really integrated in the RNA. And the second thing that we also demonstrated, that especially with the head temperature, that the poly-A tail was disappearing, mainly due to the head stress here.
So you're sure that the heat is the causing factor, or are there anything else maybe induced here, like oxidation or any impurities that can cause the degradation?
I guess that also during the IVT process, there are so many other impurities. I also saw some paper describing that some metal, divalent metal can also have an impact on the degradation of the polytail and also on the cap. And those impurities can also be created during the IVT process manufacturing. So
and
actually,
maybe it's accelerated during the heat that you induce, right?
Yes, exactly.
Yeah. Okay. So I think these questions, and I think the heat stress conditions that you chose, like the exact conditions, they are also in your paper, in your publication?
Yes.
Yes. So I think this is when someone wants to dig deeper into the conditions, I think this is where you can go in. Then there are some questions around the multimers. So first, what's the biological impact of RNA multimerization? So if you have an RNA multimer, what can this be? Can this do to the patient when it's used, for example, as a therapeutic or vaccine?
That's something that we still need to investigate. That's something definitely that needs to be investigated, but there are some papers that are actually describing the effect of these dimers, of this double strand that can have an impact on, they can lead to some immunogenicity. So depending on what you have in your aggregates, but that's something that we still need to investigate and see also if it's like, it will also, depend on if you want to use, how you want to use your mRNA, if it's more like a critical raw material or if it's more for mRNA vaccines. So that's something that needs to be more investigated in the future, for sure.
Okay, thank you. And maybe a follow-up question to this, when we talk about multimers. So you talked about immunogenicity that might be caused, but do you think that there is like a certain level, or is this also still something that needs to be discovered that is still acceptable or not acceptable?
Actually, that's a really good question. For now, I have no clue on that. And this is definitely, I mean, as for antibodies and, for antibodies, method developments, we are always trying to see if there is like an additional peak when you're developing a method that you absolutely want to characterize and see at what level it can have an impact on the patient. Here, we demonstrated some technologies to be able to monitor all the different kind of impurities. So now in the future, the question will be what exactly would be the biological or like whatever, what can be the impact of these kind of different impurities, right? And so right now I do not have the answer for that question, but that's something that definitely we open some doors with these different technologies. Now we need to go more in details and what can be the impact of these different impurities.
That's great. So analytics first and then seeing what it causes. Yeah, thank you. Then there has been a question around the denaturing conditions in ZGE and MCE. Are they the same or did you do some variations here?
The denaturing conditions are pretty similar between the two technologies, so that's why we also compared in parallel these two technologies.
Okay, great. Yeah, so the other question is again around the multimerization. So is the RNA multimerization done with itself? So the RNA is doing it to itself, or are there sequences that might induce also multimerization that it can cause more frequently?
That's also a good question. Actually, we also find an interesting thing that here it's also depending on what you have, during the IVT process, because we also saw that especially for the mRNA that has high level of aggregates, it was also associated with high level of residual solvents. So meaning that, and it is also proved that the residual solvents, especially ethanol and isopropanol, can also have an impact and leads to some aggregations. So that means that this potential multimeric form can be due also to some residual solvents.
Okay, thank you. Excellent. And we talked about multimers and poly-A. Now we come to the other analytics you have done to double-stranded RNA. So how far can you be sure that what you see, the extensions that are double-stranded RNA, not non-looped back extensions?
So for actually for the double strand, we'll just go back to one of my slides here. So to answer to the second question, so the 3' loop back, it has been demonstrated that in literature that's mainly the covalent form is represented by this 3' loop back extended, and because this one is mainly the main covalent form, when we are working under denaturing condition, you are sure that you are removing every other non-covalent secondary structures. For example, all the double strand such as dimer or this kind of thing. So for this one, we are almost sure that these are the 3' extended loop back structure. And for the dimer one, here obviously there are some investigation that needs to be done. But if you're taking a look here on this slide, you see the monomer and the dimer. Here for the dimers, the mass photometry is able actually to detect exactly twice of the size of the monomers, meaning that these are actually some dimers. But we need more investigation, obviously, just to be 100% sure about what these dimers are.
Okay, thank you, Axel. Then there is a question, is there a way to reduce non-covalent aggregates during the process, or how can you prevent this during the production of mRNA?
But that's the purpose of this paper, which is first to ready to characterize all the different impurities by developing some analytical tools, then to understand what exactly they are. And that means that right now we do not have the question. We are just like giving some clues to people. But I guess with what we did, it will help to better understand also the manufacturing process. So for example, I just talked previously about the, for example, for the poly egg tail, it can come from metals, but it can also come from residual solvents. So here, this paper is also to better characterize what we can do in terms of manufacturing processes, and maybe by letting people know that they have to be careful about the solvent that they are using, or if they are like a specific, if there is a specific step where you are going to use this solvent or these divalent metals or something like that. So these are first clues about what we can do to maybe improve the quality of the mRNA, I guess. But yeah.
Thank you, Axel. Now we reach the official end of this webinar. So this means 1 hour is over. There are some more questions in the Q&A that we couldn't cover, but we covered a lot, I think. And many, many thanks, Axel, for answering this question. I thank everyone for joining this webinar. And yeah, we recorded the whole session. And this webinar will be accessible on demand after some time. So you will receive an email from RNA leaders with an access link to get access to the on demand. You can dig deeper. You can look into the paper. And if there are any questions to Axel or to anyone here, also to Roche CustomBiotech or RNA leaders, please reach out. We are happy to stay in touch. So Jade, you want to also have some final remarks?
No, I just want to say thank you to you both for a great session. Thank you for everyone for joining. Yeah, great to see so many questions coming through, so we didn't have enough time to answer them all. I did kind of just put in the chat both of your direct email addresses. So yeah, we'll also just forward on those questions so that you guys can answer those directly afterwards. And yeah, we can also forward any of those questions along as well. So thank you for a great session. And yeah, we'll send a link from about the 23rd of April for you to put access online. So thank you both, and thank you everyone for joining. And we'll hopefully see you again virtually soon, or at one of the upcoming events later this fall. Thank you all.